Immunity. To study immunity, first, what is immunity must be known. What is immunity? Immunity is the defense mechanism present in all vertebrates which help us to fight against as well as protect against any foreign substance. Immunity is the defense mechanism shown by all vertebrates to fight and protect against any foreign substance. In case of human beings and mammals, the immune response. So what is immune response? The responses means the actions taken by the defense mechanism is called immune response. Now what are the types of immunity? Basically two. One is innate and next one is acquired. Now as I was saying, immunity is shown by all vertebrates, but in case of mammals, specifically in human beings, it's much more developed and specific. Now coming to what is innate immunity. It is inborn immune response shown by all vertebrates to same extent against all foreign agents. Now what is acquired immunity? It is developed during exposure to foreign substances and during and second exposure against same antigen shows greater intensity of response. Now from this only, from the definitions only, we can talk about the differences. So innate immune response, if I write this one is innate and here it is acquired. So first point, number one, inborn. This one, number one, developed with time after birth when exposed to different pathogens. Now what is pathogen? Disease causing organisms. What is antigen? Any foreign protein which is harmful or which can cause disease in human beings. Now this one, specific intensity of immune response against for every exposure. Every time when come in contact with a specific antigen or a foreign substance or pathogen, this intensity is same. After first encounter second exposure against same foreign substance or pathogen shows greater intensity of response. Now third point, not much. When first line of defense, innate immunity is also called first line of defense. When first line of defense falls, then acquired immunity can protect us against foreign substances. Now coming to the detail. Types of innate immunity. Basically, the types are the parts of innate immunity, the parts of our body, internal organs involved. Basically, these are external parts, or few structures are present. Basically, these are barriers. First one, physical barrier. Second one, physiological barrier. Third one, cellular barrier. And fourth one, cytokines. Now coming to detail. Physical barriers are basically the skin, also the mucus, the hairs present on surface. So what is the role of skin? We know skin is made up of epidermis and dermis. Those are intact part of our body, and which prevents entry of any living being through skin. So skin prevents entry of any living being, and also hairs and mucus trap the any organism trying to enter in our body and throws it out of it. For example, in our nasal passage, hairs as well as mucus is present inside the nostril. So whenever anything trying to, any substance, for example dust particles or pollen grains or any other pathogen, tries to enter through the nasal passage, hairs first try to throw it away, swipe it away, we can say, sweep it away, and mucus traps it and throws it away in form of sneezing. Now coming to physiological barriers. Acids present in the stomach, that is HCl, then alkaline condition maintained in small intestine, then mucus released in several parts of our body, as well as the sweat released by skin, do not help growth of any microorganism. And also saliva in our mouth. In saliva, lysozyme is present, which breaks down the cell wall of any microorganism. Now coming to cellular barriers. Leukocytes, which are a type of white blood cells. For example, polymorphonuclear leukocytes, PMNL, neutrophils, those are, and also after that it's macrophages as well as monocytes, that is natural killer cells. Actually, monocytes, polymorphonuclear leukocytes, natural killer cells, these are types of leukocytes or lymphocytes, as you can say, are present in blood, whereas macrophages are present in tissues. All of their role is to kill microorganisms by phagocytosis. What is that? That is trapping or eating. First trapping the microorganism, any foreign pathogen, foreign particle, then breaking it down into several pieces. Now what is the role of cytokine? Cytokine barriers. Virus infected cells secrete a protein called interferon, which protect non-infected cells from further viral infection. Now coming to types of acquired immunity. It is two types, that is humoral and cell mediated. Basically, humoral is controlled by antibodies and cell mediated by T lymphocytes. Now coming to what is acquired immunity, that is discussed earlier. So first we'll discuss with humoral immunity. In our body there are bone marrow and the lymphoid organs which produces antibody. Now what are antibodies? Antibodies are specific proteins which are specific proteins produced by humoral immune response, basically by bone marrow cells, and maturation is done in lymphoid organs. So antibodies are proteins produced by lymphoid immune response in response to a specific antigen. Basically, antibodies as well as antigens both are proteins and they have specific structure. Now shape of antibodies, it's Y shaped as you can see in this image. Now structure of antibodies that I'll discuss in short. As you can see in this image, there are two chains. One is light chain, another one is heavy chain. These are made up of polypeptide chains and bound together by disulfide bond. They are connected together also disulfide bond. These are disulfide bonds. Now as you can see, this smaller one is light chain and the larger one is heavy chain. And at the top of this Y structure you can see a specific sequence is there, or specific shape like lock and key, that is antigen binding site. Each and every antibody have a specific antigen binding site. Means according to the structure of antigen, antibodies are produced. Now we can see that B lymphocytes, basically B lymphocyte synthesizes antibodies, and that light chain heavy chain is represented as they have two light chains, two heavy chains, it's represented as H2L2. And there are 5 different types of antibodies produced in human beings: IgA, IgM, IgE, IgG. So these are 4. One more is IgD. So what is this Ig? Ig stands for immunoglobulin. Just a moment. IgA means immunoglobulin alpha. So Ig stands for immunoglobulin. That is another name of antibodies. Now as I told, what antibodies basically do, they recognize, or according to a specific antigen, B lymphocyte produces a specific antibody. Now they come and bind with the antigens and then they help break down using cell mediated immunity, that is CMI. So T lymphocytes are responsible for cell mediated immunity. How it is done? Basically they are produced by T lymphocytes, as I told you, and there are several proteins called major histocompatibility complex present that help in recognizing any foreign particle and destruction of it. That's why tissue grafting and transplantation, you all have heard that tissue heart transplantation, kidney, liver transplantation, these are transplanted, means from one human being, heart or kidney, from one to another person, it is transferred. Basically, transplantation means transfer of any organ in one person. If it's failure of these, due to that, transplantation can be done. For this, cell mediated immunity plays very important role. Also in tissue matching, blood group matching, also done before graft or transplantation, because our cell mediated immunity, that is T lymphocyte, tells us which is foreign and which is, that is non-self, and which is self particle or substances in our body.